रोहित जी प्लीज चेक करिए डॉक्टर रमन ने ज्वाइन किया है या नहीं किया है।
मैम उन्होंने एडमिनिस्ट्रेटर के नाम से पहले ज्वाइन किया था, अब तो नहीं है। या शायद नहीं, अभी तक तो नहीं है। मैं एक कोर्स बना दिया था, लेकिन वो है नहीं अभी। अ, या तो सर, यू कैन अनम्यूट योर सेल्फ इफ यू हैव ज्वाइंड।
हमारे सर से नहीं
Um,
आती, दो ही का आता है।
just 2 minutes, Ankur ji.
ठीक है,
I
ठीक है
just
सर,
came
ठीक है,
back from
ठीक है।
another meeting, so I just need to set up my this thing, okay? Camera
ठीक
and
है,
everything.
ओके सर, ओके ओके। अरे
Ankur ji, I'm right now ready.
Sir, as soon as 200 participants will join, we'll start.
Okay, madam. I'll wait, ma'am. No issue, madam.
Okay, sir. Okay.
नंदिनी, आप संभालना, ठीक है? मैं रेलवे स्टेशन पे हूँ।
Okay, sir. Okay. It's all handled. रोहित जी मैं विजिबल और ऑडिबल दोनों हूँ।
यस यस मैम, यस मैम।
Okay. So sorry for the late, sir. A very, very good evening to everyone.
आपका
Welcome to the
वीडियो वीडियो
day
ऑन
20.
कर। ऑन था, वो ऑफ हो गया, एक बार ऑन कर लीजिए। या डन। तो फिर से। या इट्स वर्किंग फाइन फाइन फाइन।
Working.
यस।
Okay. So good evening to everyone. Welcome to the day 26 of the ICR UGC Winter School Bharat 2025. Our winter school is an intensive 30-day national program as well as international program designed to bring together leading scientists, educators, policy experts, and young researchers from across India on one common platform. Every day we learn, we grow, and move one step closer to our objective of strengthening India's agriculture research ecosystem through capacity building, interdisciplinary learning, and meaningful discussions. Over the past 25 days, we have been privileged to host some of the finest minds in Indian agriculture, and today is another remarkable day in this journey. So, it is my privilege and honor to welcome an exceptional scientist, visionary leader, and one of the most respected names in rice biotechnology, Dr. Raman Minakshi Sundaram, sir, Director of ICR, Indian Institute of Rice Research, Hyderabad. Sir has completed completed his BSc Agri from TNAU Coimbatore, MSc Agriculture in Molecular Biology and Biotechnology from GB Pant University, Pantnagar, and PhD in Plant Sciences from the University of Hyderabad. He has further enriched his expertise through prestigious postdoctoral fellowships at Iowa State University and Cornell University, USA, as an Indo US Fellow. Sir has joined IIRR in 1998 and since then has devoted his career to groundbreaking research in biotic stress resistance, hybrid rice biotechnology, molecular breeding, and product development through transgenic approaches. He is internationally renowned for his work in trait characterization, molecular studies, and rice improvement, leading to the development of several elite and improved rice varieties. Some of his team's remarkable contributions include well known varieties such as DRR Dhan 53, DRR Dhan 58, 59, 60, 62, 66, 75, and 76, and many more, collectively contributing to the release of more than 15 new rice varieties that are already benefiting lakhs of farmers across India. Sir has undergone intensive international training, handled more than 200 major research projects, guided numerous scholars, and published extensively with 70 plus research paper and 50 plus book chapters, bulletins, and popular articles. His outstanding work has been recognized through prestigious awards including Lal Bahadur Shastri Outstanding Young Scientist Award, CSIR Innovation Award, Hari Om Ashram Trust Award, ICR Jawaharlal Nehru Award, Rashtriya Krishi Vigyan Protsahan Puruskar, and many more. He's also a fellow of NAS, a fellow of NASI, fellow of ISGPB, and serves on several important national and international committees. Sir has also been editor in chief of the Journal of Rice Research and academic editor for leading international journals. Sir, your contribution have transformed modern rice research in India, strengthened our breeding pipelines, and inspired a new generation of scientists. Your presence today is truly a honor for all of us, and with great respect and gratitude, we warmly welcome you, Dr. Raman Minakshi Sundaram sir, to grace day 26 of our Winter School 2025. We are very grateful for your presence. Over to you. The session is yours, sir. Thank you so much.
Thank you very much, madam, and thank you for the nice and kind words. Uh, shall I share my presentation? Okay.
Yes, you can start, sir.
Okay, that's fine. Um, sorry. Mm, just give me 2 minutes. I'll just figure it out. Okay.
Visible, sir.
Yeah. Um, okay. So I'll go to my full screen in just a moment. So you're able to see my full screen?
Yes, sir.
Yes.
Yeah. Thank you very much. So today we are going to talk about a topic called designing climate resilient rice through genomic assisted breeding. This is a topic which may not be familiar to a few of you, but rice breeding is a topic which should be very familiar to most of you. So I will be describing not in terms of a theoretical lecture or class, but in terms of what we learned and what mistakes we did it, and then how we rectified those mistakes in the later years. So this is describes my journey in rice research for the last 28 years. So I've divided my presentation into about 5 parts. First, I'll be describing about the present status of rice in the country, the threats to rice production, and then possible solutions. And after that, I'll be introducing you to genomic assisted breeding. Then I'll be talking briefly about genomic assisted molecular mapping, including biparental cross mapping, marker trait linkages, genome-wide diversity, how to exploit natural allelic variants, and finally ending with genomic selection or genome-wide association study. Then I'll be talking about a few application cases of genomic assisted breeding or molecular breeding or marker assisted breeding for climate resilience in rice. Some of our own examples and then some examples from other groups also. And I'll also be briefly be talking about new tools which can accelerate genomic assisted breeding. About 3 or 4 tools I'll cover. And finally, I'll give my conclusion, my own conclusion. These need not be accurate or correct, but what I have learned in the last 28 years in terms of using tools of molecular biology and also molecular breeding and then genomic assisted breeding, what I have learned and what are all the future prospects, I'll be just highlighting them in 2 slides. What I request is that after presenting each one of these parts, I will just stop briefly, and if you have any questions or queries, I'll be happy to answer. So shall we move ahead forward? Okay, so now let's move to the first part of my presentation, wherein we'll be seeing rice from an Indian perspective. All of you are aware that rice is one of the most important staple food crops of India. It feeds about 43% of Indians and then takes care of their calorific requirements. Presently, the rice area is somewhere around 45 to 47 million hectares, almost about 22 or 23% of the total cropped area in the country. But for many of the rice farmers who need to eat daily rice for their food and nutrition, rice is life. So as far as the production situation is concerned, this year, that is 24-25, we produced around 149 million tons of rice. And we also exported about 20 million tons of basmati and non-basmati rices, earning more than 80,000 crores or more than $1 billion worth of foreign exchange. But what we need to see is that what we produce right now is very, very good. And we have almost achieved about 3.2 tons of rice per hectare on an average across the country. And then certain states like Punjab, Haryana, Telangana, and Andhra Pradesh produce almost about 5 to 6 tons of rice per hectare. Certain states are still lagging, like parts of Bihar, Assam and other states are still lagging behind. Orissa is also lagging behind. But overall, the present situation is very, very satisfactory. But what is you need to understand is that for the last 2 decades, we didn't see any major yield gain or yield jump in rice. Rice yields are almost plateauing, or the productivity levels are plateauing somewhere between 2.2 to 3 tons per hectare. And what we possibly need to understand is that our requirements or food production requirements by the year 2050 is about we need to produce 168 million tons of rice to feed at least 1.62 billion people. And this rice has to essentially come not from 45 or 47 million hectares. It needs to come from 40 million hectares or even 35 million hectares, because bulk of the area under rice is being converted to other crops. So we need to produce more from less of area. This is the important take-home message from this slide. Now coming to what are all the major challenges with regard to rice in the country. One of the major challenges I would like to mention here is that even though our population growth rate is not as serious as earlier yesteryears, but still our population is steadily growing at a rate of around 1.3 or 1.4%. And what we are also seeing is that rice yield gains are not very, very significant in the last 10 years or 15 years. Rice yields are plateauing, and there is also a threat of rapidly changing climate, and I'll come to that a bit later. And that's a real threat. And rice is both a causative agent for climate change or methane gas emission, and also a victim of climate change. And because of a changing climate, we are also seeing more and more minor pests and diseases getting converted to major pests. And other important issue is that unlike wheat or maize, wherein the crop is grown mostly in farable ecology, rice is grown mainly in a farable ecology like irrigated ecology or rainfed lowland ecology. It's also grown in other unfarable ecology like upland, hills and others. I'll come to that later. And another important concept which has been floated by our Honorable Prime Minister is that he aspires to consider rice as not only a crop which fulfills our calorific requirement, he also wants to rice to satisfy the nutritional requirement like micronutrients, proteins, and other essential nutrients. And we need to develop rice varieties which are not only high yielding, but also which can fulfill our nutritional security needs. And finally, the land, water, and then other resources are shrinking. Input costs are rising, and the money available for rice cultivation for farmers, the profit margins are rapidly decreasing. So these are all the problems. I would like to highlight one of the most important problem, that is climate change. We all know that rice grown under submerged condition can cause significant emission of greenhouse gas called methane. Rice grown under dryland condition emits some limited quantity of nitrous oxide. So rice is one of the causative agents resulting in about 3% of the total greenhouse gas emissions from agricultural ecosystems. But what we need to understand is that rice is also a victim of climate change. And we all understand that global annual mean temperature is expected to increase by about 1.5 degrees centigrade by 2050 and 3 degrees centigrade by 2000. And already we are seeing from 2000 values that are recorded in the year 2000, the present temperature has already increased by 1 degrees. And possibly by 2030 itself, temperatures may increase by about 1.5 degrees. And each 1 degree increase in mean land temperature, physiological studies have shown that rice yields could decrease by 10%. Another important impact of rapidly changing climate with rising global temperatures, emission of greenhouse gases is that there will be increased periods of drought, salinization, and most importantly, increased incidence of pests and diseases. And if you look into this particular map of India, these are all rice growing areas, and most part of India, particularly the western part, central part, are all, like say, highly vulnerable to changing climate. Red ones indicate most vulnerable, green ones indicate less vulnerable. All the high productivity ecosystems in Andhra Pradesh, Tamil Nadu are all highly vulnerable. And this even evident from this particular map, which shows that certain parts of India, particularly central part, western part, and northeastern part, are very, very highly vulnerable. And if we don't take adequate steps to address the issue of climate change, the total rice production may significantly decrease. And added to the climate change factor, I mentioned that rice is grown both in favorable ecologies and unfavorable conditions. Favorable ecologies include irrigated rice, which is being grown in about 56.3% of area and gives about 3 tons or 3.5 tons of production or productivity. But the other ecology, rainfed lowland ecology, it's moderately good. The production under rainfed ecology is marginally lower, and it is even lower in ecologies like uplands, hills, and then below sea level. And rice is also the crop that is being grown from 2500 meter elevation in snow-capped Himalayan peaks. Also in Kashmir Valley also it is being grown. It's also being grown in areas in Kerala wherein the land is below sea level. So it's also being grown in saline soils and also acidic soils, and even under submerged conditions also, deep water also, rice is being grown. So because of these unfavorable ecosystems, our country's overall productivity is low as compared to other countries like China, USA, Egypt and all, wherein rice is being mainly grown in favorable irrigated ecology. And we also need to understand that because of climate change, from Green Revolution era, that is starting with 1965, we had about three major insect pests: yellow stem borer, garbage, and then green leaf hopper. As on date, the number has increased to about 20. This year, one more pest has come into the picture called rice root knot nematode. And similarly, with regard to disease scenario, during Green Revolution era, we hardly had one or two major diseases like brown spot and then blast. But this year, we are noting that there are 12 major diseases, and some of them have assumed epidemic proportions in several parts of the country. Now, coming to what all the tools available for developing climate resilient rice. Some of you may be breeders, so you understand that conventional breeding will be helpful to develop varieties, but it has got limited scope because it takes a lot of time, and it's so I'm sorry. I'm sorry. I'm having a very bad cough and cold. Excuse me. So what we need to understand is that conventional breeding, the application part is very, very limited in terms of quickly developing climate resilient rice varieties. Marker-state selection or molecular breeding is a very good tool. I'll talk about some of the products or some of the applications of molecular breeding. Equally good are tools like genetic engineering, which has got regulatory issues, and gene editing, which has got excellent scope, but still IPR issues remain. And genomics could be one tool which can not only increase the efficiency of conventional breeding, but also molecular breeding. and even genetic engineering and gene editing also will be benefited by genomics. Now, am I clear about the first part of my presentation? Shall I move to the next part? Okay. Now let's see what is exactly genomics or genomics-assisted breeding. Genomics is the science of studying the genome sequences. So it has got two parts. One is structural genomics, in which we read the sequences or we study the sequences. And second part of genomics is functional genomics, in which we'll be studying the function of each and every gene or each and every sequence and try to interpret what gene it encodes or what trait it encodes. So genomics assisted breeding is nothing but combination of genomic tools and marker assisted breeding. So the actual definition, as indicated by Dr. Pallavi Sinha, says that genomics-assisted breeding is the integration of genomic tools for improving the efficiency of plant breeding. So genomics-assisted breeding leverages advancements in structural and functional genomics to identify genes or alleles associated with desirable traits and accelerate the transfer into new rice lines. And the key components of genomics-assisted breeding include molecular mapping or QTL mapping, which we'll be seeing in the next session. Then we'll also be seeing something about marker-assisted selection, then genomic selection, allele mining, and then genome editing finally. Then if you look into the steps in genomic-assisted selection or genomic-assisted breeding, is that trait selection and then phenotyping, then genotyping, identifying marker trait linkages through molecular marker analysis, and also genome-wide selection association study and validation of the markers, then utilizing this marker information through SNP chips or some other high throughput tools for rapidly developing high value breeding lines through population improvement and also trait pyramiding, then their field testing and validation, and finally multi-environmental trials leading to their release and commercialization. Now, let's look into the innate aspects of genomics-assisted breeding. What are all its components? The first and foremost component of any genomics-assisted breeding effort is the availability of genetic resources. Without genetic resources, no genomics-assisted breeding cannot progress. So genetic resources include core collections, donor identification, biparental mapping populations, diverse unrelated panels, which form association panels, then multiparental derived populations like magic populations or NAM populations. These are all the basic tools used for doing genomic analysis of the genome. Then we have second part of it called trait discovery, in which we'll be doing linkage mapping, QTL-seq, and also association mapping. And this will result in availability of large set of PCR-based markers, and most importantly, sequence-based markers like single nucleotide polymorphism markers, whole genome resequencing based markers, and genotyping by sequencing markers. And some of these markers have also been converted into chips, wherein a chip will have all the traits that are present in rice, and these chips can be gainfully used. Then after working with genetic resources and doing the trait discovery, we go for trait improvement, in which the various steps include marker-assisted selection, marker-assisted backcross breeding, marker-assisted forward breeding, marker-assisted recurrent selection, gene pyramiding, multi-trait introgression, genomic selection, and also haplotype-based breeding. Then now let's look into applications of genomics-assisted breeding for rice improvement. So as we all know, this is a simple equation which is called as Breeder's equation or genetic gain equation. And this equation says that genetic gain is directly proportional to the selection intensity, the kind of intensity with which you will be doing the selection. It's also directly proportional to the selection accuracy. And most importantly, genetic gain will occur only when genetic variation is there. And genetic gain is inversely proportional to the time taken or generation interval. So genomics assisted breeding can address the genetic equation by increasing the breeding efficiency, or by directly by making the selection intensity faster, or reducing the generation interval, but also making accurate selection of desirable traits. It can also add precision to our breeding process, wherein when we will be doing backcross breeding, it will minimize the introduction of undesirable traits from donor lines. And finally, overall genetic gain will be enhanced by identification combination of desirable traits more effectively, leading to faster genetic improvement. And finally, genomics assisted breeding is the only tool through which we can rapidly develop climate resilient varieties. This is simply because this is the fastest tool that is available. Now, I finished my second part of my presentation. Are there any questions or comments you would like to offer? There are no comments. Let's try to dive deep into how genomics is assisting or enhancing our capacity to do quick molecular mapping. So initially, all of us use biparental cross-derived mapping populations to map important traits. For example, in the year 2006 and 2008, one major bacterial blight resistant gene was identified by the group at PAU using microsatellite markers, which are not very dense across the genome. Then, just within, say, about 6 years, using new tools like sequence-based analysis, they were able to identify the candidate gene for this particular gene called XA38, and they were also able to land on that particular gene. But the process took from course mapping, identifying the location on a particular chromosome, that actual identification of candidate gene, and then develop sequence-based markers. It took another 6 years. Then another example is the effort by Central Rice Research Institute, wherein they identified that there is a very promising donor called as Salkati, which has got strong BPH resistance. Through genetic analysis, they identified two major QTLs on chromosome 3, and then chromosome 4, and then they identified that these two QTLs are very nearby. But for actual identification of the candidate genes underlying the major QTL, QBPH 4.3, it took them another 4 to 6 years. Then similar is the effort with our own story, wherein with the help of SSR markers, we identified a major bacterial blight resistance gene from Oryza nivara through biparental cross-derived mapping population. But for us to identify the candidate gene, it took another 3 to 4 years. Same is the case wherein we have identified a candidate gene for BPH resistance from a breeding line called RP2068. So from this line, we identified a gene called as heat shock protein gene, but the whole process took from main gene identification, by molecular mapping to gene identification and then trait interrogation. It took to about 2 to 3 years. But the scenario rapidly changed when sufficient genomic information was made available, when SNP chips were available. So when we initially identified a major bacterial blight resistant gene located on chromosome 11, long arm of chromosome 11 from Oryza officinalis, which is a wild rice, then we deployed a 7K SNP chip, which helped us to identify the candidate gene within about 1.5 years, wherein we could identify the major QTL also very, very precisely. Unfortunately, the SNP marker was lying on a candidate gene which encodes NBRC domain containing protein, which was derived from the wild rice. Similar is the story wherein one of my colleagues, Dr. Seshu Madhav, he identified a major neck blast resistance gene from Oryza glumipetla, which is another wild rice. So from trait discovery on course mapping on chromosome 3, he was able to quickly, within about 1.5 years, he was able to identify the candidate gene and then develop candidate gene specific SNP markers, which are being used in breeding. Another recent example is that we were able to identify a major QTL associated with sheet blight tolerance from a landrace called as Vazopec. As you can see here, Samba Masuri is totally killed by sheet blight disease, but Vazopec stands very, very tall. And my colleague Dr. Prakasam, in consultation with Dr. Gopalji Jha's group, he deployed genotyping by resequencing strategy, wherein the mapping population was resequenced, and he was able to precisely identify not only the major QTL, but also the candidate gene associated with that. And he was able to quickly design SNP marker, which could distinguish the two alleles very, very easily. And this took only 1 year from SSR or microsatellite based course mapping to fine mapping and candidate gene identification using SNP markers. Another typical example of association mapping is done by my colleague Dr. Jyoti Badri, wherein she has utilized a set of cultivars, around 100 lines which are having various levels of culm strength or stem strength. And using this particular analysis, using genome-wide sequencing of all the 100 cultivars, she was able to identify major and also minor QTLs. And she was able to identify one major QTL located on chromosome 3, which she was able to utilize in breeding programs. Now coming to genomic selection, that is one important topic wherein I would like all of you to focus a little bit carefully. Generally, genomic selection is not routinely used for crop breeding in public sector breeding system. It is routinely used in some private sector breeding for crops like maize, tomato, and all. And it is very, very routinely used for breeding for higher milk production or higher meat production in cattle. So what they do is that first, they will make a cross between the initial parents and develop half-sib lines or test crosses, and they'll be carrying it forward to stable generation, and they'll be then training they'll be sequencing all the offsprings or all the recombinant inbred lines derived from the half-sibs or lines or test crosses, and based on the results what they are getting from the stable population, then they'll be making out fresh crosses, and they'll be analyzing how much is the market rate prediction, which is being done based on both genotyping and phenotyping. That is called as genomics estimated breeding value, or GEBV. So based on GEBV value, which is constantly revised based on the prediction models, they'll be using large set of genotypes for as parents and also for developing better crosses. Remember, in genomic selection, we are not bothered about what is the candidate gene underlying the markers which are showing high GBV or high genomic estimated breeding value. We are only bothered about how close they are there to a particular trait or particular this thing. We are not bothered about candidate gene discovery. So genomic selection centers around the genomic estimated breeding value of each and every SNP marker which we are discovering. So as I mentioned earlier, this genomic selection, unlike other methods like marker-assisted backcross breeding or forward breeding, which is generally effective for improvement of oligogenic or monogenic traits, this genomic selection is very, very effective for traits controlled by polygenes or multiple genes, each having small effect QTLs. Here we are not bothered about what genes these QTLs code. We just, based on marker trait correlation, we just use them for breeding program. Then this is effective for identification of superior parents or segregating progeny, and this information can be utilized in forward breeding. And we need to simulate several crosses and also say several this thing, and there is no one fixed model which we have to use it for genomic selection. We need to use various models. I'll explain about one model which we have developed. This will optimize the genetic gain what we'll be receiving. Genomic selection, if your training population or test population is very well designed and you identify very good marker trait linkages without bothering about what gene it encodes, then it will be very, very useful, and it can, within a time period of 3 to 4 years, it can result in development of many improved breeding lines. And what is important is that with application of tools like speed breeding and also artificial intelligence, we are working more towards integrated genomic selection, wherein high throughput tools like computing, speed breeding under controlled growth conditions, and information from bioinformatics or data points are used for accelerating in genomic selection. And I'll give some example how we are doing genomic selection at my institute, that is ICR, Indian Institute of Rice Research. We have a large set of population which have already been genotyped. These lines we are using the genotype versus phenotype information for model development, and we are using these particular models or populations which have already been genotyped and phenotyped. Based on that, we'll develop model, and using this particular models or marker trait associations, we will be advancing lines which have got which we predict to have higher genome estimated breeding value, and we may cross with those lines or parents having high GBV value, and based on that we make crosses and advanced generation, and we develop elite breeding lines. So all these are crosses which involve elite into elite lines, particularly in genomic selection. That's what we need to understand, and this will help us to develop varieties in a faster manner which not only have one or two traits improved for multiple traits. And one such model which my team is using for genomic selection is a model called artificial neural network based genetic algorithm wherein we use for predicting grain yield under low nitrogen conditions. So we have got a plot which have got graded levels of nitrogen. So we are identifying genotypes which can give yield at varied levels of nitrogen, a property called as nitrogen use efficiency. So for analyzing the nitrogen use efficiency associated markers or the genomic regions, we use this particular model where it's a statistical model. How it was developed, it is actually described here. So this was able to predict very accurately with almost 0.79% R square value or correlation value very accurately. Remember, no genomic prediction model, genomic selection model is 100% efficient. Each model has got its false discovery rate. And till date, nobody has created the perfect genomic selection model. For example, one important thing what we are deploying is that genomic selection is being deployed very widely in our All India Coordinated Research Project on rice, wherein in the year 2023 and 24, all the lines that were nominated were fingerprinted with 1K RICA SNP panel. 1K means that has got representation of more than 1000 genomic regions, and it is a moderate throughput SNP platform. So we are using that to predict which center is nominating diverse lines, which center is nominating low diverse lines. We are also using this information also to predict loci or SNP markers which have got high GEBV and which can be used for breeding programs. And now I'll just talk about I'll stop here with regard to genomic system molecular mapping. Is there any questions or comments? Okay, now I'll move to one of a few applications wherein we have used molecular markers and genomics for development of climate resilient rice. I'm sorry. As you all know, biotic stresses are a major production constraint of rice. They limit rice yields up to 30%, which means this year if we have produced 149 million tons of rice, we could have actually harvested more than 180 or 190 million tons of rice. About 30 to 50 million tons of rice, we have lost it to insect pests and diseases. With regard to insect pests, the major ones in rice are stem borer, leaf folder, BPH, and gall mitch. With regard to diseases, they include blast, bacterial leaf blight, sheet blight, rice tungro virus, and then false smut. Nematodes and weeds and are also problems which are very severe. Among the insect pests, resistant sources are available only for BPH and gall mitch. For stem borer and leaf folder, till date we have not got a strong resistant source. So molecular breeding or genomic assisted breeding is not possible with regard to stem borer resistance or leaf folder resistance. But for breeding for durable BPH resistance or gall mitch resistance, it is quite possible. And with regard to diseases, like bacterial blight, blast, and up to some extent sheet blight, we now have genetic solutions wherein strong resistant donors are available and molecular breeding is possible. And initially resistant breeding started way back in 1970s, wherein single gene containing donors were widely used. But with the changing climate and increasing temperatures, what we are seeing is their single gene containing varieties are breaking down fast in terms of their durable resistance. And with the changing climate, we need to deploy at least two genes or two QTLs or more in their combination through gene pyramiding or gene stacking. Now let me describe about our own work with regard to how we made Samba Masuri bacterial blight resistant. As you all aware, Samba Masuri, which is also known by the trade name Sona Masuri, it's a very popular variety in South India, which has got moderately high yield, and it also has got excellent grain and cooking quality. For example, in the states of Andhra Pradesh, Tamil Nadu, Karnataka, and also in Telangana, as compared to other varieties, Sona Masuri or Samba Masuri as it is called, the original name is BPT 5204. It commands a premium price of at least minimum 500 to 1000 rupees per quintal extra. But unfortunately, this variety is highly susceptible to all the known pests and diseases and also to all abiotic stresses. And when I joined IARR way back in 1998, a group of farmers who are growing Samba Masuri, essentially from coastal Andhra Pradesh, they approached my institute telling that we are getting this the particular disease-like symptom. What is this disease? They wanted us to identify, and they also wanted some solution to manage this disease. So we later did a thorough analysis and we identified that the disease symptoms are matching bacterial blight. Then we isolated the pathogen, re-inoculated, and established that this disease is caused by bacterial pathogen called Xanthomonas oryzae, PU oryzae. And then later we try to identify the most effective gene combinations. We identified that XA21, 13, and 5, these three gene combinations, when combined in the background of Samba Masuri, they'll be effective. So we used a donor parent called SS1113, which was there in the background of a Punjab variety called PR120, PR106. And that was used for transfer to Samba Masuri through marker-assisted backcross breeding. We had specific markers targeting XA21, XA13, and XA5. These markers were used for selection for plants containing these genes at each backcross generation. And we also used a set of SSR markers, about 90 markers, which are polymorphic between the parents SS1113 and Samba Masuri. They were used for background selection or to quickly identify the backcross derived plants which are having more contribution from Samba Masuri. And backcrossing was carried out till BC4 generation. And by BC4 generation, we identified a plant having all the three genes in homozygous condition, and that plant was selfed, and then uh, that particular plant was selfed, and then a series of BC4F2 lines were developed, and we identified 5 lines having all the three genes in homozygous condition, and also with maximum recovery of Samba Masuri genome. And that particular line was then later released as a variety. This line, breeding line, was known as IT19046, which had all the three genes. And also this line was very closely looking very similar to Samba Masuri. So this line was later tested in All India Coordinated Research Project on Rice Trials, notified and then released as a new variety by Government of India. So this is a success story wherein we didn't do it alone. We did it together with a basic sciences research institute of CSIR called Center for Cellular and Molecular Biology. And as on date, improved Samba Masuri is being grown in more than 1 lakh hectares across the country in several states. And this variety has been licensed to several private seed companies on a non-exclusive basis. We are receiving significant royalty revenue also, and it yields the same similar to Samba Masuri. Its grain and cooking quality are also similar to Samba Masuri, but it has got durable bacterial blight resistance. And surprisingly, it still has got about 2% from the donor parent. And the donor parent is a known low glycemic index rice variety. Recently, in 2017, we also identified that improved Samba Masuri is also a low glycemic index rice variety. Low glycemic index indicates how much of glucose is released after consumption of say 100 grams of cooked rice, while most of the rice varieties like Samba Masuri, Swarna and all release about 62 to 65 grams of glucose within 3 to 4 hours after eating rice. This variety releases only 51% or 50.99%. So this has been classified through clinical trials to have a low glycemic index. So there is a renewed interest among farmers to grow this variety and consume healthy rice. And if anybody is interested, in this particular paper, we have described the nuts and bolts of how improved Samba Masuri was developed from Samba Masuri. They can go ahead and this is how a crop of improved Samba Masuri looks. And what is important is that whenever the disease occurs, this particular variety, improved Samba Masuri, as you can see in this picture, is already started maturing and it's full of grains. While Samba Masuri, particularly this was taken in a location near Kakinada, which was affected by a cyclone in the year 2017. We had a big cyclone called Hudhud, which affected several parts of coastal Andhra Pradesh. So Samba Masuri was totally devastated. As you can see here, you cannot even see a single grain bearing panicle. All are affected severely by bacterial blight disease, while improved Samba Masuri has already matured and then it is reaching the senescent stage and then it's perfectly free from bacterial blight. And a survey done by Dr. Amarendra Reddy from National Academy of Agricultural Extension Managers estimated that those farmers in East Godavari who grow improved Samba Masuri, because of reducing loss due to bacterial blight, they are gained in just 5 years about 240 crores. A recent survey says that the net gain from growing improved Samba Masuri in bacterial blight prone areas is more than 2,400 crores. Consider this with the cost that was involved in developing improved Samba Masuri. It was hardly about 48 lakhs, which was given to both CCMV and Hyderabad total. So this I'm trying to show it to you just to tell that macro-assisted selection or genomics-assisted selection, even though some cost is involved, the benefits are manifold. But when we developed improved Samba Masuri, we didn't bask at our glory. That we wanted to ensure that Samba Masuri is not only protected against bacterial blight, against other pests and diseases, and against a few abiotic stresses also. So we developed more improved versions of improved Samba Masuri by adding value to it, by adding major genes to it. That was possible only through genomics-assisted breeding, wherein initially we developed a variety called DRR 53, which had two additional genes in addition to 21, 13, and 5, called XA 33 and 38. Then that variety was released as DRR 53. Then later we released another variety called as DRR 60, which has got, in addition to 21, 13, and 5, it also has got blast resistance called PA2 and PA54. And we are nearly ready in terms of developing varieties with BPH and Galmage resistance. As far as abiotic stresses are concerned, we have transferred a major QTL conferring tolerance to low soil phosphorus, which means the variety can grow under even 40% reduction in phosphatic fertilizers like DAP or single superphosphate. So this variety's name is DRR 60. I'll show you some good pictures of DRR 60. And we also released a variety called DRR 58, which has got a major QTL called as Saltol. And Samba Masuri is mainly grown in coastal Andhra Pradesh, coastal Tamil Nadu, and then coastal Karnataka. So by developing DRR 58, we are addressing coastal salinity problem also. And recently, we transferred some of the yield enhancing genes and developed, we transferred genes associated with high zinc and developed DRR 48. Remember, all these derivative lines like DRR 53, DRR 60, DRR 62, 58, all are low glycemic. But my colleague Dr. Neeraja, she has transferred some genes associated or QTLs associated with high zinc and developed DRR 48. It's also doing very well in farmers' field. And finally, one major product what we have developed is a high-yielding version of improved Samba Masuri, which not only has bacterial blight resistance, blast resistance, but also has got yield-enhancing genes, a gene called as OSSPL14, which increases the spikelet number per panicle, another gene called as SCM2, which increases the culm strength, or it makes the plant lesser prone to lodging. We are also developing submergence tolerance versions and drought tolerant versions. Remember, we are not trying to put all the traits or all the lines, all the genes into the background of Samba Masuri or improved Samba Masuri. Depending on the requirement, we are just adding one or two traits or three traits into the background of improved Samba Masuri. So these are the products I would like to tell. So this line is practically immune to any known races of bacterial blight because it packs a punch. It has got an additional gene called XA38. And since we didn't go for backcrossing till BC3 or BC4, we just stopped at one backcross because the donor parent also was in a fine drain type. We are able to identify the segregants which are 10% higher yielding as compared to Samba Masuri. And for those who want to know about this development, people can refer to this particular paper. And my colleague, Dr. Srinivas Prasad, developed blast-resistant versions, which are nearly immune to blast disease. As you can see here, this is DRR 60, which is indicated in yellow arrows. And it's totally immune to blast disease, while improved Samba Masuri and other entries are highly prone to blast disease. It's also resistant to bacterial blight also. And DRR 58 is another example wherein we used genomic tools, high throughput genomic tools, and we used SNP markers both for foreground and background selection. And this was found to out-yield the recurrent parent improved Samba Masuri by 27% and the tolerant check by 5% in coastal salinity trials. And this line is doing really well in farmers' fields and also in experimental plots which are put for demonstration. For example, this is one of the inland salinity plots at Trichy, Trichurapalli, Agriculture College, Trichurapalli, which is prone to high levels of inland salinity and alkalinity. Samba Masuri literally gets killed or burnt, while Dehradun 58 is still surviving. For those who want to know a little bit more about this development, they can refer to our paper, which was published in the journal Rice. Then another example of a abiotic stress tolerant variety is Dehradun 60, which has all the three resistant genes XA21, 13, and 5, and a major gene called as PUP1, which encodes a PS-tol kinase. And this variety is now presently being grown in more than 90,000 hectares across the country. And as you can see here clearly, um, improved Samba Masuri under low phosphorus conditions, or when you apply only 60% of phosphatic fertilizers, it doesn't grow really well. And the introgress line, that is Dehradun 60, grows really well under low phosphorus conditions. And Dehradun 58 and 60 were dedicated to the nation by our Honorable Prime Minister, Shri Narendra Modi ji. And Dehradun 48 is another very good example wherein genomic tools we could transfer high zinc QTLs from CSR 27. So this line, we have estimated that it's not only resistant to bacterial blight, but also tolerant to salinity. And it also shows a very good levels of zinc in the polished grain, about 24 ppm. And another important point is that this also has got low glycemic index. So a company called Glymino is marketing this rice called as Glymino rice under their own brand name with a co-branding. Then another example is that wherein genomics has really helped us to develop a better product is the development of Dehradun 75, which is a high-yielding line of improved Samba Masuri, which not only has the necessary resistant gene, but also has got yield enhancing gene called as SCM2 from a Japanese rice variety called Habataki, and gene called as Wealthy Farmers Panicle or OSSPL14, which enhances panicle branching. As you can see here, as compared to Samba Masuri or improved Samba Masuri, the introgression lines possess higher yield, and this variety in this year, particularly in coastal Andhra Pradesh and also in Telangana, there were heavy rains during crop maturity period. Most of the other varieties have lost, but this variety, which almost under normal condition yields 6 tons per hectare, when the farms are managed very well, farmers have even recorded and then shared videos of this variety wherein yields of around 10 tons, 11 tons have also been shown. If you're interested, just type Dehradun 75 in YouTube. You'll find that there are a lot of reports about superior performance of this variety and Stampede at our institute, wherein we had about, during the start of this Kharif season, we had about 12 tons of seed. We thought that will be sufficient. That was distributed within half a day. And finally, within 3 day period, after a lot of stampedes, we managed to distribute about 20 tons of seeds of this variety. And similar work we have done in another variety called MTU 1010, wherein the panicle sizes have been increased by transferring these genes. Then I would like to highlight a few other varieties, not in the background of Samba Masuri, but in other backgrounds, which have got certain climate resilience, which we develop through our marker-assisted breeding or genomics tools. One is the world's first saline-tolerant hybrid called DRRH5, which has got medium slender grain type and salinity tolerance. Dehradun 52, which has got terminal heat stress tolerance. Then another recent example is a variety in the background of the very popular variety called Krishna Hamsa, which is the Rabi variety. We have transferred cold tolerance, drought tolerance, BPH resistance, and also blast resistance into background of Krishna Hamsa through convergence breeding. And the product has been tested under normal trials and then released as a new variety. And another typical example wherein my colleague Dr. Neeraj has significantly contributed is for submergence tolerance. Working at IRRI, she has developed a submergence tolerant version of Swarna called as Sub1, Swarna Sub1, which tolerates submergence up to about 12 to 15 days or 20 days. And then it grows really well when the water recedes well. Another example is Dehradun 42, which is a NIL in the background of IR64. It's really tolerant to drought. You can see original IR64 is totally killed, while this particular entry is surviving very, very coolly under severe drought stress. And this has got two major QTLs called QDTL 2.1 and 3.1. And another important example is application of genomic assisted selection is development of durable bacterial blight and blast resistant varieties, which occupies more than 30% of the total area. And recently, under the able guidance of my guru, Dr. A.K. Singh, they have not only improved basmati varieties for bacterial blight and blast resistance, they have demonstrated that basmati varieties can be grown under dry DSR conditions, saving water and by saving lot of cost for the farmers in terms of manual or mechanical weeding. We now have got herbicide tolerant basmati varieties wherein a mutant gene called as robin has been transferred. And if you look into the overall picture, it is estimated that climate resilient rice varieties occupy at least 25% of the total area under rice. In the last few years, we have released more than 44 numbers of climate resilient varieties and hybrids, which are occupying about 10 million hectares. And each of them have a yield advantage of 1 to 1.5 tons per hectare. And these are earning an additional revenue of more than 10,000 crores. Now I'll briefly talk about, with just 4 slides, about the new tools available to accelerate genomic assisted breeding. The first and foremost tool is called as haplotype based breeding. We all need to understand that for each and every trait, there are not just 2 alleles. We generally consider there is a favorable allele favorable for the farmer or for the breeder, and non-favorable allele. That's not the case. There are several favorable alleles and several non-favorable alleles. By combining the different favorable alleles for different genes, that is basically called as haplotype. So haplotype based breeding essentially arises from large scale genomics effort wherein several important agronomically important genes have been discovered and their alleles have also been discovered. And based on the superior combination of alleles, which is called as haplotype, and their variation, we are now able to develop superior rice varieties containing the favorable haplotype. For example, my colleague Dr. Jyoti, working closely with IRRI, she is working on several favorable haplotypes associated with yield, grain filling, and also grain type and grain size and shape. And she's trying to develop superior varieties combining multiple genes through mark-resisted forward breeding or pedigree breeding. Now, speed breeding is another game changer. In speed breeding, what we do is that generally rice in North India can be taken up only for one season in a year. In South India, possibly we can take up 2 or 2.5 seasons in a year. In certain parts of South India, we can take up to 3 years. But under speed breeding, wherein we control the day length, rice, as you all know, we know that it's a short day plant. And by increasing the light intensity or day length, by increasing the temperature or manipulating the temperature, by adjusting the humidity, and by harvesting the seeds in immature conditions, and then putting them in a hot air oven to fast mature them. In crops like wheat and rice, we can grow even 6 generations in a year. For those who are interested, you can visit ISARC or IRRI South Asia Regional Center Varanasi, wherein they are having a very good speed breeding center. In my institute also will very shortly develop the speed breeding facility, okay? And we'll be demonstrating it. And speed breeding is not only suitable for rice, but also from wild rice also it's very, very highly suitable. And another important tool is genome editing, where in conventional breeding, if you want to manipulate a particular or introduce favorable allele for a particular gene, you also introduce a lot of non-favorable loci associated with the genes, which are indicated here in orange color arrows. Then through genetic engineering, you can precisely introduce a favorable gene, but genetic engineering has got its own disadvantage in the sense that it may not go and then replace the original locus. It may go and then bind to some other locus, and then that problems are there with regards to regulation and biosafety. But genome editing is a tool. We don't introduce any foreign gene here. Only the known gene that is mutated, one or few sequences are mutated, and other than that, there is no change in the background genome. And gene editing is being done through various strategies like TAL effector nuclease mediated gene editing, zinc finger protein mediated gene editing. Of late, the most popular method of gene editing is CRISPR-Cas9 or CRISPR-Cas12a mediated gene editing. And this gene editing approach is very, very fast, and product delivery is possible within 3 to 5 years. And it can be one of the future of plant breeding. And that we have demonstrated it by manipulating a gene which is associated with cytokinin biosynthesis or cytokinin degradation. This gene is called as cytokinin oxidase, which degrades cytokinin. By targeting this gene, my young colleague Dr. Satyendra, taking cues from the original inventor, he has edited two exons of this gene called as exon 5, it's exon 3 and exon 4, and he has developed a higher yielding and climate resilient version of the popular variety Samba Masuri or BPT 5204, which is not only higher yielding with larger panicles, because more cytokinin means more panicle branching and then higher yield, but it also has got early maturity, but it has got the same kind of grain and cooking quality like Samba Masuri. As you can see here, this line is being able to grow under increased conditions of drought, low nitrogen, and also it can grow very, very fast even under low nutrient conditions. This is one application. And last but not the least, artificial intelligence is turning out to be an important tool wherein it can be used for your germplasm panel selection in terms of fine-tuning our phenotype predictions. It can be used for handling big phenotype data, sequence data, and phenomics data, sensor data. It can also be used for biological interpretation, making the marker trait associations quick and then fast and then precise. It can also be used for using the available data, marker trait phenotype associations. It can be helpful to develop without making any crosses. We can predict how far this crosses will be good enough. And finally, it will help us to design new varieties in a faster manner and then more precisely. For those who are interested to know more about artificial intelligence applications in plant breeding, they can refer to this particular book. I'll shortly share my slides or presentations in PDF form with Dr. Ankur and Dr. Nandini. Feel free to distribute it to the participants. There is no IPR associated with that. Feel free to use the slides also in your presentation. And there are various applications of artificial intelligence. I would not like to name them, each one of them. And now the approach has gone from genomic assisted breeding 1 to genomic assisted breeding 2, which integrates tools like speed breeding, gene editing, pangenomics or panomics, super panomics, de novo domestication, and artificial intelligence, and then big data. And that review paper, which I indicated here, has got complete information about all these things, how each one, how they are able to develop superior crop varieties. Now, I would like to conclude my presentation. First and foremost thing, for development of any good product, we need to work closely, not only interdisciplinary work, interinstitutional work is also important. For example, for development of improved Samba Masuri, I work closely with my breeders, with my molecular biologist, with my pathologist, with my agronomist, and also soil scientist. And we also work closely with another institution called CSIR, Center for Cellular and Molecular Biology, or CSIR CCMB. And this collaboration even now also continuing. All the products which I showed, like DRR 53, 60, 58, 62, 75, are all products developed through this collaboration. And we need to understand that improved Samba Masuri in the last several years has resulted in increased revenue every year to a tune of about 1,200 crore rupees to the farmers. And what we need to understand is that we identify the right set of genes for deployment, that is XA21, 13, and 5. And later, through DRR 53, we added another gene called XA38. So right selection of gene combination is important. And we also need to understand that just out of an investment of about 48 lakhs, we gave a lot of benefits to the farmers in terms of our this particular project through which we developed improved Samba Masuri. And just transferring one or few traits are not important. It is important to pack multiple traits through genomic assisted breeding or through genomic selection in order to develop climate resilient rice. And genomics has grown really well, but what has not grown really well in our country is functional genomics, that is understanding the function of each and every gene. And it is certainly our country needs to invest more in terms of identification of greater number of economically important genes and alleles which are relevant to India. And we need to give more funds for functional genomics research. And finally, new tools and techniques like gene editing, speed breeding, haplotype-based breeding, application of artificial intelligence, machine learning in breeding opens a window of opportunity for improving of recalcitrant traits, particularly many of the climate resilient traits are recalcitrant. It is very difficult to manage them through molecular breeding or through conventional breeding. But one important thing I would like to, it's a thought for you. You can just go through these slides and then see whether I'm talking is making sense or not. I'm claiming that rice production in India is climate resilient for the past few years. This I'm talking with hardcore data. If you look into the rice production from 2001 to 2014-15, whenever we had a off year, for example, in 2001, 2, it was monsoons were very good. So rice production stood at 93.3 million tons of milled rice. But when 2002-3, when monsoon rains failed, we had a drought period. So rice production dropped down. That was the story. Every time monsoons failed, rice production dropped by marginally or drastically. But starting for the last 5 years, I'm proud or happy to say that rice production, irrespective of failure of monsoons or reduced rains in rice growing regions, rice production has steadily increased. And this year we are received record rice production, which is almost equivalent to that of number one country, that is China. And possibly by the year 2030 or 45, we'll be producing more rice in lesser area and more nutritious rice also. And I would not like to claim full credit, but works of farmers who are hardworking, adopting new varieties, that has made a lot of difference in terms of adoption of climate resilient varieties. Equally important is the hard work done by ground level breeders working together with molecular biologists like me or molecular breeders like me, and their work should also be acknowledged here. And finally, as a breeder, imagine that a breeder can be equated with a mechanic. Mechanic has many tools. Among these tools, genomic tools like biotechnology are only one among the many tools available to a breeder. Remember, a mechanic cannot work with a spanner alone. He has to sometimes work with a cutting player, a screwdriver, and all. So, biotechnological tools cannot replace breeding. They can only complement and supplement the process of breeding. That's a point I want you to take care forward or carry forward. Biotechnology tools can only help a breeder in terms of improving his breeding efficiency or selection efficiency and develop varieties or hybrids in a faster manner. They cannot replace breeding. Sorry. Sorry, I've got a bad cough. So these are all my colleagues who have helped us to all this work. This is not my individual work, it's a team work wherein Dr. Satyendra has worked towards application of molecular tools for rapid trait discovery. And he is also the source or janani for genome edited rice, that is ZRR-than 100 or Kamla. Dr. Neeraja is working on nutrient use efficiency and also biofortification. Dr. Divya is working on wild rice introgression. Dr. Fiaz has got an ambitious project called Target 1000, wherein he's trying to develop rice with at least 800 or 1000 filled grains per panicle. With just about 5 to 8 panicles, he thinks that we can meet the food requirements by high grain number rice varieties. Then Dr. Girish is also working on wild rice introgression. Dr. Jyoti is working on constant and plant type related traits. Dr. Kalyani is working on root related traits. Dr. Paparao is a molecular biologist who's working on durable biotic stress resistance. This is my team, and thank you for your patient listening. And I now stop my presentation here and thank the organizers for providing this opportunity.
Thank you so much, sir, for the wonderful session. Even if you are sick and your health isn't all right, still you gave the session. Thank you so much. If anyone is having any kind of question, you can either unmute yourself or you can type your queries. I will read it out. So please.
शुभदीप जी बताइए. Okay. This is something
Hello.
very subjective. His question says that what genomic evidence can help predict whether, yeah, sorry,
Someone is speaking,
Shubdeep
I
ji,
think.
yeah, Shubdeep ji has got a very interesting question. What genomic evidence can help predict whether a gene edit will have pleiotropic effects in rice metabolic pathways? What we generally do is that when products will be finally be released. Still, you need to understand that Kamla has been dedicated, but still it has been notified and then released by our CVRC. So what we are doing is that we are undertaken a complete metabolome profile of not only the growing rice plant but also the grain. And what we have observed is that because of higher accumulation of cytokinin levels, even at a young plant stage, the photosynthetic efficiency has been increased, and that pushes the plant more towards accumulating the necessary photosynthetases and then maturing early. That's one of the reasons for DRR than 100 to mature early. But remember, this early maturity is not phenomenon wherein we can observe it everywhere. For example, when you grow Kamla in Kashmir or probably in a cooler climate, it may take more time or equivalent time as compared to Samba Masuri in terms of maturity period. But with regard to the pleiotropic effects, we are trying to understand by in terms of whole genome metabolic analysis of not only the growing plants but also the grains. Also, we do at grain filling stage, and also after polishing, we undertake a whole genome metabolic profiling. We do undertake, and already we have completed the proximate analysis of both Samba Masuri and also DRR than 100. Hope I'm clear with my answer, Shubhadeep ji. Thank you. So,
Thank you, sir.
yeah, thank you. And one question from Dr. Telam Ratan Singh is that please elaborate a bit on implications of A in plant breeding. Just like A has got tremendous capabilities and also limitation and capability for misuse, it applies to plant breeding also. For example, in terms of we are doing our All India coordinated rice improvement project trials every year using AI, one of the AI tools, home developed by my colleague. We have actually identified certain centers suitable for certain ecologies wherein stability of performance is seen. So we are now shortened the number of centers, drop those centers wherein year-to-year variations are seen, and we are now able to identify or limit the number of locations. And using AI, we are also able to predict which parental combinations can give better hybrid just by seeing their SNP genotyping. These are all few examples, but I'm not an expert on AI in plant breeding. What I'll recommend is that there are two papers, one by Tyagi et al. And another by Yadav et al. So I'd like you to go through that paper and then try to understand a little bit more. And if you have any specific queries further, can always email me. My email is there in the beginning of the presentation. How do differences between Japonica and Indica rice genomes influence the choice of CRISPR targets for specific traits? Remember, even though it's not 100% necessary, gene editing will be better if we have got the complete sequence of the target gene which we are editing in the target genotype or the target variety. Without that information, it is just like shooting in the dark. For example, when we did whole protein analysis, three-dimensional analysis of the protein cytokinin oxidase in the original variety Habataki and also in Samba Masuri, what we've observed is that we try to fit in cytokinin in the cytokinin oxidase enzyme. In the 3D structure, we observed that exon 3 and exon 4 are more relevant in terms of giving better reduction in degradation of cytokinin. So it all depends on whether your variety has got some amount of drafts genome sequence information. Even if it is not available, through designing some PCR primers targeting your gene, you can amplify it, study its sequence, and then design the targets. Without understanding or knowing which target gene you want to edit and what is the genotype you are going to edit, I will not recommend you to go for gene editing. I'm not an expert in gene editing. I'm just only a facilitator. Dr. Satyendra will be able to answer, and he's also a sweet person. His email ID is skm Satyendra Kumar Mangrotia. मतलब इसका initials है skmdrr@gmail.com. skmdrr@gmail.com. You can ask your questions to Dr. Satyendra, taking the talk as reference. He'll be able to answer your questions with much greater depth. And one more question is, can AI predict how a crop variety will perform in future climate scenarios at different geographies? Yes. provided we got proper AI tools to develop. And there are various prediction models that are being developed in terms of predicting if temperatures increase by 1.5 degrees centigrade, what kind of varieties we require, what are all the traits that are required. If temperature increases by 3 degrees, what are all the genes or what are all the major loci that are required. Predictions are going on. It's a work in progress. I cannot tell with certainty or with practical example, but AI can certainly predict how crop variety will perform in future climate scenarios or different geographies.
Thank you so much, sir. And sorry, that was my question only.
Yeah.
As we are also working on AI integration and all that, and
Mm.
we are from Hyderabad itself. If you permit us, probably we can come and meet you,
Yes,
as
ma'am.
we are
Can
doing a lot of
certainly come.
R&D on that.
Yeah, yeah. Certainly, I don't apply AI tools, but I can
Yeah.
connect you with a group based at IRRI South Asia Hub here in ICRISAT. Dr.
Okay.
Pallavi Sinha, who is our own PhD student. After PhD, she joined International Rice Research Institute South Asia Hub. I can connect you with her. Just send me an email. I will pass on her contact details and then you can take it forward. Because when I say we are using the tool, I'm not an expert in that. Always take expert advice before designing any plan. So Dr. Pallavi is helping us to make sense out of eCrypt data.
Right, sir. Thank you so much. I'll send an email.
And Karpagavalli has asked a question. Can you receive resistant source of germplasm for rice BLB? Certainly, if you are from a public sector organization, we'll give it to you free of cost or for a small processing fee. Any donor line, whatever that is available with us, we can pass it on. Even for private sector, there are some formalities. If your company has got a foreign major shareholding, then you have to get a permission from National Biodiversity Authority. And after that, we can give it to you for a small cost. I'm not sure whether you work in public or private sector. Accordingly, our strategy will also differ.
Good evening, sir. Sir, please provide Dr. Satendra mail ID again, sir. Please, sir.
Yeah, it is SKM. Is initial Satendra Kumar Mangrotia, SKM. DRR at gmail.com. SKM DRR at gmail.com. And
skm@gmail.com.
yeah, skmdrr at gmail.com. There is no dot in between. It is skmdrr. Our institute was earlier known as, prior to 2014, it was known as Directorate of Rice Research. Probably Satendra must have opened his account prior to 2014. That's why his email ID says that skmdrr at gmail.com. So one question from Avinash Prabhakar Karjuleji.
Thank you, sir.
Yeah. Is there any possibility of use of gene editing for improvement of seed storability? This is an important prospect because one of the major problem what we see in rice seeds is that if it is not properly stored, it gets rancid and then it gets spoiled very quickly. One tool what we are trying to knock out is a gene called as lipases and lipoxynase group of enzymes. We are trying to knock it out, and this is indeed a certain possibility. We means not our own group, but some group located in Philippines are trying to knock them out. That is indeed possible. So which variety of rice is more suitable for consumption with respect to health support? If you ask me, I'll say you can safely consume DRR than 48 or improved Samba Masuri. But your preference depends on which region you come from. For example, if you are from South India or from Maharashtra, possibly you'll prefer rice which is fine grain type, short slender or medium slender. So in that category, we have got a few varieties which have got low glycemic index and then high grain zinc. If you can write to me, I can suggest these varieties to you and then tell where they may be available. Some of them are also available through Flipkart and Amazon. But I need to know what is your preference. Accordingly, I can suggest. I think we are done with the questions.
Sir, good afternoon, sir.
बताइए.
Sir, what is the possibilities of training or internship in your institute, and how can I approach that? If you
एक
could
हमारा
clarify
वेबसाइट
that.
थोड़ा देख लीजिए. काफी हम लोग वो सोशल मीडिया में भी एडवर्टाइज करते हैं. पिछले साल हम लोगों ने एक पेड प्रोग्राम कंडक्ट किए थे जिसमें काफी सारा प्राइवेट सेक्टर से भी ऑन पेमेंट ऑफ अ नॉमिनल चार्ज पीपल पार्टिसिपेटेड. फॉर ऑल पब्लिक सेक्टर पार्टिसिपेंट्स वी मेक इट फ्री ऑफ कॉस्ट. थोड़ा आप किस एस्पेक्ट में आपको ट्रेनिंग चाहिए, मुझे ईमेल लिख दीजिए, rmsudram34@gmail.com। इसमें मेरे प्रेजेंटेशन के बिगनिंग में भी है, उसमें मुझे ईमेल लिख दीजिए, आई विल रिस्पॉन्ड। ओके।
Sir, I am a student, so that's
सो
why I
स्टूडेंट
am
है तो
asking.
आपका कोई
Currently, I am
चार्ज
in
नहीं है,
BTech second year.
just you have to take care of your accommodation and then your transport charges. Training will provide it free of cost.
Okay, sir, I will email you. Thank you, sir. Means
हेलो
a lot. Thank you, sir.
Thank you.
हेलो सर।
बताइए।
हेलो सर।
Yeah, tell me.
सर, sir, we are from coastal part of this India, sir, like from Odisha and from part of West Bengal. Sir,
Okay.
we have some problem of production of fine rice. Sir. Till now, some fine rice is demanded here. We are not getting good varieties, sir.
Sir, you write to me.
Sir, here you should
Better
have some.
than 58 will suit your coastal salinity ecosystem. It's a medium slender
Yes,
grain
sir.
variety with a low glycemic index, so you can grow it. And coastal salinity system, we also have a lot of bacterial blight. So I would recommend you to go for
Yeah.
dearer than 58, sir. Just write an email to me. I'll take it forward, sir.
Okay, sir. Thank
Small
you.
quantity, we'll send it free of cost. It is up to 2 kgs or 5 kgs. Just give me your address, we'll send it free of cost.
Okay, sir. Okay.
Good evening, sir.
Yes, tell me, madam.
Actually, I'm working at CAU Infal. So, can we get some A line and B line from your center?
Okay, we can get it, ma. Actually, what you need to do is send an email to me telling that you want to start a new breeding program regarding hybrid rice. You need some promising parental lines and you want to assess
Yes,
their
sir.
suitability for infall conditions. We can certainly provide at least 10 15 male parents, that is restorer lines, and at least three or
Okay.
four pairs of A and B lines we'll be happy to provide. We'll be happy to do it. You are a
Thank you,
faculty
sir.
here or you are a student here? Yeah, go ahead. If you are a faculty, then we'll be happy to provide
Um,
immediately.
I'm faculty.
Yeah, so we'll be happy to provide. And your center is one of our
Okay, sir. Thank you,
voluntary
sir.
center. Earlier, Dr. Ranjit Sharma used
Yes,
to
sir.
handle our trials. Right now, I'm not sure who is handling our
Yes, yes.
trials, but you can send an email.
One.
Yeah,
Okay, sir. We'll send.
uh,
Okay.
send
Thank
an
you very
email
much.
to him. I will certainly send it to you, ma. Okay,
Okay.
thank you. Any other questions or queries? If there are no further
If
queries,
anyone wants to ask any question, Sir is himself answering everything. Please ask the question and also you can type. Okay, that's all, sir. So we'll end the session by the vote of thanks. Thank you so much, sir, for being here for an exceptional and enlightening session today. Your lecture gave us deep insights about complex scientific processes and everything you have shared with us: biotechnology, molecular breeding, trait characterization, and the real impact of improved rice varieties on farmers across India. Your simplicity in explaining everything, the process, the passion for research, and your dedication to strengthening India's food security truly inspired every participant today. We especially appreciate the way you shared your journey of developing the landmark and the rice varieties, the challenges behind hybrid rice improvement, and the future scope for the young researchers in biotechnology and crop improvement. Your words have not only expanded our technical understanding, but also motivated us to think innovatively and work with commitment towards nation building. On the behalf of Winter School Bharat 2025, ICR, and all the participants, we express our heartfelt gratitude to you, sir, for your valuable time, your guidance, and your contribution to Indian agriculture. We look forward to learning from you again in the future academic activities. Please be with us in the future also. Thank you so much.
thank you very much, madam. Thank you for the opportunity. I will shortly convert my presentation into PDF file because PowerPoint is almost 200 MB in size, so I cannot be not be able to share it through email or WhatsApp. I'll convert that to PDF file and then share it with Ankur ji so
It's
that he
okay,
can
sir.
freely
It's
distribute it to the participants. Okay. Thank you very much.
okay, sir.
Okay. I'll log out
Okay, okay.
now.
Thank
Okay.
you so much, sir.
Thank you. Thank you,
Yes,
everyone.
sir.
Thank you.
Everyone can leave. Thank you so much for today.